So my name is Hossein. I'm co-founder and CTO of Lucid Genomics. Lucid Genomics is a spin-off of Max Planck Institute and Charité Hospital. We are a team of people with different backgrounds, from biologists, geneticists, computer scientists, machine learning expert, and medical doctor teamed up with technology as expert in software to build a software which analyzes the whole genome and make the whole genome actionable. Talking about whole genome, I need to kind of introduce a little bit of biology, maybe 01, and it's about, the DNA in each cell is like a cookbook of the cell. So in this cookbook, you have the recipes of proteins, the genes which are translated to proteins, and these proteins are acting. If you look at the DNA, you see only 2% of these whole cookbook is translated to proteins, even less. And there are regions between these genes in the DNA which are called non-coding DNA or dark genome. And this is for a while being unknown. What is the role of these part of the genome? Making the whole genome actionable means that we want to bring the knowledge and our understanding from non-coding DNA into action. We want to diagnose more diseases by integrating of non-coding and coding together. And what we know, at least for type of diseases called rare diseases, which 400 million, 350 million people around the globe, they have this kind of disease, which is diseases, which are 7,000 types of diseases. We can diagnose only 40% of them. And we don't know why we cannot beyond the 40%. You may ask, how sure are you that this non-coding elements will contribute to this, for example, at rare disease diagnosis, this 60% undiagnosed patients. So we know that there are many functional elements in non-coding DNA. For example, enhancers, promoters. There are also elements which are shaping DNA in 3D. If you look at the DNA, it's like a thread, 2-meter long thread in one cell, which is clustered, so to say, in chromosomes. Chromosome 1, 2, 3, chromosome X and Y. And if you look at it linearly, one way is look at it linearly, where you say there are these genes in this chromosome, but you have to look at it in 3D because these thread gets folded. And there are many elements in non-coding DNA which are affecting or ruling out how these elements are, how this thread becomes a 3D shape and how it regulates, how this 3D shape regulates the gene expression. So the question is now how much we can go further by understanding the non-coding DNA. And the answer is we don't know it yet, but we know that we have proof of concept scientifically that these elements have functional roles, and also we know that mutations in non-coding element cause diseases. Already we have many cases, and also we know that 95% of, for example, a large-scale studies, we found mutations in non-coding element which are associating, not causing, but associating with different traits. So the signal is there, but the data and the technology was not there to answer the question. And now the technology is available. The sequencing price dropped from a couple of thousands, for example, 5 years ago, to $100 recently. And no one asks now, shall I sequence my genome if I have a genetic problem? So that's not the case anymore. Then the question is how to analyze it and make best out of this genome to answer my biological question. And since the data is now available, we can answer these questions at scale because we are dealing with very complex with biology. It's a very complex system, and understanding this complex system needs to bring every layer of information one can add, and one of these layers is reading out this cookbook first, then trying to understand how this cookbook may affect gene expression and integrate all these data together to answer our questions, not only in diagnostics, but also in other fields. So having this data available, we see different changes in the ecosystem. For example, one change is, at least we know in diagnostic, we use now the whole genome to diagnose more patients. We have biobanks that at a scale sequencing whole genome data to understand the biology behind human DNA. But human is not the only organism which has DNA in the body. If you look around, DNA is everywhere. And people may only think about, when we talk about genomics, analyzing DNA, we only talk about health and disease. But also I can say that understanding the DNA, reading out the mutations in the DNA, which at least in human body is like 4 million different types of mutation, which makes the difference between me and everyone. Going beyond health and disease in humanity, we can ask many questions by looking at genomics. For example, the Nobel Prize in 2022 went to Leipzig, Max Planck Institute. I think Professor Pabo got the Nobel Prize, and it was because of his work on Neanderthals and sequencing the genome of Neanderthals and understanding the human history. So analyzing DNA, genomics, has broad applications, which we are only focusing on one of these applications for now. And it's very important to couple it with AI because of different reasons. One reason which we integrated in our product as our first technology is that the current data that we can read from human DNA or other organisms' DNA is very noisy, and detection of mutations in such a noisy input data is very tough. Here, AI can come and help. It doesn't matter if it's a DNA of human, healthy or diseased, or, for example, an animal, it's a horse DNA. We produced an AI model which can find the signals which are leading us to the existence of mutations in the genome. So with AI, human eye and the algorithm, without AI, human eye and the algorithm cannot find these signals, but AI can detect that and tell us, hey guys, there is a mutation here that you may not see that, and then we can take that mutation and ask ourselves, what's the role of this mutation? So this is one part of it, and that's in the denoising the data. We can if we can use AI to denoise the data. The other part is interpreting and understanding these mutations. What does that mean? We are looking for the patterns in available patient genomes, which might help us to answer the question of a new patient being undiagnosed, and we look at those mutations which have the same signal and try to diagnose not a patient with even another type of disease. And here AI can help also, can help human and biology, I would say, very strongly. Journey has started from 2021. Before that, we were also we were scientists at Max Planck Institute working on the same topic, and at that time, our question was, looking at the new technologies, reading out the whole DNA for understanding coding and non-coding, what would be the future technology? And after a while, we understood that the dominant technology, which is again very noisy, is going to be the future one. So what we did was using AI for denoising of the current technologies and the most dominant technology, using the more accurate ones, so and less scalable and more expensive ones. So this was an idea that it came to our mind during our work at Max Planck Institute, that scientifically, like, we have resources to use the more expensive, more accurate one, but at the scale, we need to change the gear and use the current data and the dominant one. And this was, I would say, the first step towards Lucid Genomics, aside from the interpretation of the detecting mutations. And this is what at least I'm very proud of. And from there on, we worked on many unsolved cases, and we found candidates that, with our tool product as a whole, not only talking about detection part, but also interpretation part, that helps us to diagnose more patients. So this is our current day-to-day job that we have. We are working on cohorts of patients' data being unsolved, some of them for 10 years. And we already found a couple of good candidates that we are close to say, yes, they're solved and they're diagnosed with our tool. And we are so proud of these cases, which has to be proved in future soon. About healthcare and how it would look like in 10 years, I think, at least this is not my point of view, American Journal of Human Genetics published that most of the human genome, will be sequenced by 2030, and they have their whole DNA in their gadgets. So it's a big claim. I don't want to claim that. I see also, like, we are dealing with sensitive data. But what I see also from myself is, like, the fear of having your genome in the gadgets is reducing, because at the same time we see the value that it can give us through this journey, through my life, from looking at my DNA. And whenever you see as a community, when we see that the benefit is much higher than the risk, then the change will happen. And of course, like, these changes may not happen in more conservative communities, but if these happen in some other country, then the other will follow, and they see that, okay, the value is there, the benefit is already can help the community, then the people's mindset will also change during these years. So I think we are going towards personalized medicine. We need to know to whom we are prescribing something. And to whom means that what's the DNA, what's the cookbook of that person, and how we can integrate the difference of someone, for example, with Chinese background, with someone with European background. So these are the important elements that has to be integrated in our healthcare, in our diagnosis, and also in our prognosis, and of course treatments. So I would say integrating the cookbook of all cells of my body in my healthcare is essential. Yeah.
Good.
Talking about scaling up or tailoring the treatments towards minor populations can also come from here. That, for example, the drug discovery companies, if they focus on one population, or they focus on the bigger population or the reference that they have, Now they can also focus or understand why some drugs are failing, knowing that the background of these patients is something else, and how they can tailor drugs towards different backgrounds. And about ethics in genomics, I would say it's super hard topic. A friend of mine from Australian National University contacted me 2020, and we were talking about genomics, and he has background in ethics, and we were discussing a lot about topics like because I really want myself to sequence my DNA, and even I'm happy to share it with the others. At this part of my life, of course, but I ask my question, if I share my DNA with the others, do I need a consent from my brother or not? Do I need a consent from my parents? And ethics in genomics is very important, because it's not about only me as a person, but also about relatives and the risk factors and the risk that every relative of mine with the same genetic background may face. So at current state, I would say that in Lucid Genomics, we know that we are dealing with sensitive data, and of course in Germany we know that the sensitivity of analyzing this. So at the same time, we know the value of democratizing the genomic data for the others. For example, sometimes you find two people with same background, but they're in different continents, and just having these two people with the same disease, you can diagnose both of them at the same time, while before knowing these two patients is not possible, because different mutations maybe in the same gene can help us understanding what was the cause of genetic disease for these patients. So it's a very sensitive topic. At the same time, we need to democratize it. And we see, at least in higher level, there are these movements in not only Germany but worldwide to bring the infrastructure that at the same time it secure data of the patients, but also enable the power of democratizing it for better health for everyone. So at Lucid Genomics, what we do currently is we deal at every patient's data as, of course, super sensitive data, should not be shared with anyone, and should be only used for the purpose that the data is available to us. And yeah, that's it. Okay.